Great, thank you. I'm really pleased to be here to introduce Dr. Brooks Fu, who we recruited, I guess, a little over a year ago. Brooks has an outstanding background, having done his graduate work at Caltech with invasive intracranial neurophysiology in human volunteers studying what we call error-related signals and understanding how the brain recognizes errors. And then he continued his work further with Uli Rudishauser, who's one of the thought leaders in our field in intracranial neurophysiology, and did further work on computational modeling of neuronal networks related to cognitive control of our motor intents and other, not just motor, but beyond motor control. So he's come here now to build out our intracranial human neurophysiology program, part of our human neurosciences program with the Donald Brain Institute, working collaboratively with the neurosurgeons, but also helping with recruitment of other people to really enhance our footprint in the space and really doing outstanding work. So I'm looking forward to having him meet everyone and present his work. Thank you.
Thank you, Nader, for your kind introduction, and thank you everyone for being here for my talk today. So in my talk today, I'll focus mostly on the progress we've made recently in understanding how metacognition works in the brain. So what is metacognition? It's our ability to think about our thinking. It's the ability for us to reflect upon our thoughts and behaviors and to really introspect. This idea of metacognition actually have very deep philosophical roots. Great thinkers throughout the history have recognized the importance of metacognition in shaping our lives and in leading us to wisdom. For example, Confucius famously said in the Analects, quote, "I examine myself 3 times every day." I believe many of us do too. But what he is trying to emphasize here is reflecting on our actions in order to ensure that we stay true to our values and responsibilities. And in ancient Greece, the famous Delphic maxim, "Know thyself," which is inscribed on the temple of Apollo, reminds us of the timeless importance of self-awareness. Both Eastern and Western philosophy have long understood that knowing ourselves is key to making better decisions and judgments and living a fulfilling life. And now, neuroscience starts to help us to review the brain mechanisms behind this ancient wisdom. In more modern and scientific contexts, metacognition allows us to evaluate our cognitive processes based on self-generated internal feedbacks. Imagine when you make a small mistake when you become absent-minded. You quickly realize it after without anyone pointing it out. This is metacognition in action. We're able to recognize when we are distracted. We're able to evaluate our decisions, and we're able even to assess how confident we are about our own decisions. Metacognition also help us to tell the difference between what's going on in our mind and what's going on around us. And this is actually crucial for making good decisions and for us to adapt in this fast-changing world. By reflecting on our actions, we can actually make improvements. We will adjust when we notice our poor choices, and we'll become more confident as things are going well. But when this ability becomes impaired in conditions such as psychiatric and neurological disorders, it creates big troubles for the patients, and it leads to significant lower quality of life. But the strange thing is, despite how important metacognition is that we still lack a very effective treatment options to rescue and restore metacognition to its normal level in these psychiatric patients. So it's especially important for us to understand the brain mechanism behind metacognition for us to be able to develop these treatment options to help these patients. So in recent decades, there is significant progress made in identifying the brain regions involved in metacognition. One key area identified is the medial frontal cortex, which is shown here as colored areas in the plot. Despite these advancements, we still do not fully understand how the neural circuitry that support metacognition works. Metacognition is complex. It happens very fast and it relies on subjective reports, and it's actually very difficult to measure. Human studies use all sorts of creative tasks to elicit metacognitive reports, but the brain measurement tools that we use in these studies are mostly non-invasive, so they don't actually directly capture neural activity. Plus, they lack the spatial and temporal resolution that are necessary to tease apart these metacognitive processes from all these other brain signals that coexist. On the other hand, invasive studies in animals allows researchers to record a large number of single neurons with high precision. But the challenge here is that the animals needs really a lot of training, and they lack the level of cognitive flexibility that we humans have. And also they cannot effectively communicate their self-awareness. So this limits how relevant these findings are for human psychiatric conditions. So in order to tackle these challenges, we have partnered with clinicians in the Cedars-Sinai Medical Center and the UT Southwestern. Together, we will record neural activity directly from inside the human brain, while these patients perform the tasks that engage metacognition. You can see an illustration of the hybrid electrodes that we use on the bottom right. Our research would not be possible without the generous participation of our patients. Our patients have drug-resistant epilepsy, which requires the implantation of electrodes to map out the seizure activity. While they sit in the hospital rooms waiting for a seizure to occur, they collaborate with us to perform behavioral tasks. This allows us to simultaneously record neural and behavioral data with the setup you see on the right. On the bottom left, you can see a stretch of data from one of these recordings. When a neuron fires, it generates spikes. And these spikes are picked up by our electrodes as shown as these little ticks in the graph. We take these spikes out and then we cluster these spikes according to the waveforms and sort them into different units. Error monitoring is a key aspect of metacognition, and much of our work so far focuses on understanding its neural mechanisms. So we ask our subjects to perform a task where they get distracted and they make a fair amount of errors. So while they're doing this, we record from the medial frontal cortex in their brain. On the left, you will see the locations of where these wires are implanted. And on the right on the screen, you will see what we call an error neuron. And let me walk you through this complicated looking plot. So this plot has two parts. The upper panel is what we call the RAS plot. Each row represents one single attempt that the subject made at the task. And each little dots here you see represent a single spike that this neuron fired. And blue dots are from attempts where subject performed correctly, but they received no distraction. Orange represent trials where subjects perform correctly, but they receive a lot of distraction. And on errors are shown here actually as the red trials. And the bottom panel actually is we take the average of these little dots in each of these conditions. I want to draw your attention to the peak here, which actually shows there's a significant increase of spike rates on error trials that happens right after the subject pressed the wrong key. The amazing thing about this signal is its timing. It occurs right after or shortly after a subject pressed the wrong key, but before they were shown any visual feedback. This tells us that these neurons, these error neurons, participate in this self-monitoring mechanism, meaning that the brain can detect and monitor these errors without any external inputs. So a defining feature of human cognition is its incredible flexibility. We humans are generalists. We're able to pick up various of different tasks, often just by following very simple verbal instructions. In artificial intelligence research, building a general AI system that can do the same, that to become a generalist is actually considered a holy grail. But even with very advanced systems built today, like ChatGPT, we're still a bit far from reaching this goal. Our discovery of error neurons in the human brain lead us to wonder, are these neurons generalists or specialists? Do the same neurons, the same set of neurons, detect errors across multiple different tasks, or do we need a specialized error neurons for each specific task? In order to explore this question, we asked our patients to perform yet another task, so 2 different tasks back to back, where they will receive all sorts of different types of destruction and where they will make errors. So what do we find? We discovered that a significant number of error neurons that will report errors across 2 different tasks. The plots shown here are pretty much very similar to the plots I showed earlier, but I want to again draw your attention to the red trace here. This red trace represents error response of this neuron. What is remarkable about this error response is that they reaches the same height at roughly the same time and with almost the same persistence. Now, I want to emphasize to you that these 2 tasks that our patient perform are quite different. They involve completely different visual details, motor responses, and task rules. So this makes these neurons, what we're looking at here, truly domain general. It does not care where the errors are, which tasks occur, it just consistently report errors. But this is not the whole story. While many neurons in our sample were domain general like this one, we also find a significant number of error neurons that carry error signal that vary between different tasks. So how do we make sense of this diversity? It's very important to remember that neurons do not function in isolation. They have neighbors. They function in a population, in a network. What we found actually is that domain general neurons like this one and domain specific neurons, they coexist peacefully in the near proximity in the brain. So now the big question is, do the signals from these domain general neurons get drawn out by all these other domain specific ones? And how do we interpret this diversity in error responses across this neuronal population in the medial prefrontal cortex? So we decided to develop a novel method to analyze our data. Here's the trick. We can think of each neuron as one in this network, in this population, as one coordinate. And the pattern of activity across all these neurons will map to locations in the high dimensional space, what we call the neuronal state space. To help with visualization, we'll use a technique called the principal component analysis to further reduce and map this high dimensional representation to a 3-dimensional space shown here. So this allows us to get a clearer and more geometric understanding of how the medial frontal cortex processes information. In this figure here, each circle represents the population response under a task condition in a given task. So here, red circles represent population response on average on the error trials, and green circles represent population response on average on the correct trials. So we connect these red dots and green dots within each task to aid visualization for you. What really stands out here in this plot is that these circles are not randomly distributed. Instead, they are actually organized in a rather specific way with a specific geometry. You can see that the black line here are roughly parallel, and the red circles occupy one side, and the green circles occupy the opposite side in a very neat fashion. So now the key question is, can a downstream neuron who listen to a bunch of these medial frontal cortex neurons still pick up a domain general error signal despite all these different neurons firing simultaneously? The answer is yes, it can. And let me explain how we see this from this plot. So in the neuronal state space, every location represents, let me remind you, a specific task condition that the neurons jointly represent. So the lines across this space actually represent coding dimensions, what we call. And the broken bar here is exactly one such domain general coding dimension for error signals. To see that this is domain general, we start by projecting the green circles and the red circles onto this gray broken bar. To do this, in order to do this projection, what we're doing here is trying to find a position along this gray broken bar that's closest in distance to either the green or the red circle. And these projected points are shown on this gray bar as green and red rings. And then we apply the same trick to the task 2 data, projecting the red and green circle from task 2 data also to this gray broken bar. What we found immediately is that the projection from the task 2 actually completely overlapped with our projection from task 1. And however, the red and green rings representing error and correct responses separately remain clearly separated. What this means to us is that along this gray bar, we can accurately separate error response from correct response, but we can no longer tell which task these rings are from, whether they are from task 1 or from task 2. So this complete removal of task-specific information means we have identified a domain general error coding dimension, a dimension that signal errors without any regard to the task itself. In fact, we can take this a step further by using machine learning methods to classify error and correct trust after making these projections. Shown here on the right is such decoding result. What is really remarkable about this analysis is that the same classifier was able to distinguish error and correct trust in both tasks with almost perfect accuracy. And these results, in summary, give us a new and geometric way to understand how generalization works in the human brain. It also offers us a working definition of domain generality, which is a complete separation between the task-specific information and the variable of interest, in this case, error detection. We found similar geometric insight also applies to the case where medial frontal cortex neuron represent the presence or absence of distractions during task performance. So this highlights the generality of our novel approach. Our current direction in the lab include honing in and refining this geometric approach to understand various types of metacognition. We're also actively investigating how our brain is able to compute confidence signals in various cognitive domains. And also, we're able to evaluate our actions, how we are able to evaluate our actions by integrating external and internal information. We also are keen to apply the insight we gain from studying human metacognition to improve AI systems. Lastly, one very key mission in our lab is to ultimately develop neuromodulatory treatment options for human patients. And I should mention that our lab is currently recruiting grad students and postdocs. And should any of this result sounds intriguing to you, please come and talk to me. I'm happy to chat anytime. Lastly, I want to thank you for listening, and I want to thank my previous lab at Cedars-Sinai Medical Center and Caltech, where a lot of this work has been done. I would also like to thank our patients and families. Without them, without their generous support, this work would not have been possible. Thank you.